Okay, Assalamu alaikum. Hello and good afternoon, everyone. I am Dr. Asma Rizwan, and I welcome you on behalf of the Academy X to this IGCSE Biology session 2025. The Academy X has been the leading academy for almost more than 10 years now across the UAE and Qatar, with more than 500 students taught in GCSE, securing amazing results. If you have any questions or information, you can always reach out to us on the number shown on the bottom right of your screen, which is 971-50318-6765. Or you can also scan the QR code right in front of you on the screen. Should you have any questions regarding the presentation or otherwise, please hold them or write them down in the chat box or with yourself, and we'll address them at the end of the session. I would appreciate if everybody pays attention to their mics so as to avoid any disturbances. Thank you for signing up for this session and hope we all have a productive time. So we'll move on with the presentation. This includes all syllabus content for all the exams, and today we'll be covering the systems of the human body. Just to give you a general idea, these are all the systems that are part of the human body. The underlined with the red are the ones that we'll be covering. So it includes the nervous system, respiratory system, circulatory system, digestive system, excretory system, integumentary system, which includes the skin, endocrine system, and the reproductive systems. So this is just a general overview of the systems and their organs. The organs actually make up the system. So we have the nervous system, which includes our brain and the spinal cord. All the body systems are controlled by this nervous system. We have the endocrine system, respiratory, cardiovascular, digestive, reproductive, the skin, and the urinary system that we'll be covering. Okay, starting with the major system, which, as I said before, is the nervous system. It controls all the coordination and response. And it includes two systems, the nervous system and the endocrine system. So all the systems are coordinated by the nervous system, and the endocrine system deals with the hormone release, which controls our internal environment in response to our external environment as well. So the nervous system it involves a stimulus, which is a change in an animal's surroundings, and it involves a response, which is a reaction to that change. So the change, for example, was detected by your eyes, which are an example of a receptor organ. So the organ is a receptor organ which receives the information, any change in your environment. The response was brought about by contraction of muscles, which are a type of effector organ. They produce an effect. The nervous system links these two and is an example of a coordination system. So the summary of the sequence of events that is occurring is a stimulus, which is received by a receptor, coordinated by the nervous system, sent to the effector organ, and that is how you respond. Okay, moving forward. So this actually is showing the pathway of the nervous system, which is called a reflex action. So the nerve pathway of a reflex is called the reflex arc. Basically, arc part means that the pathway goes into the central nervous system and then straight back out again in a sort of a curve or arc. We will further in this presentation see what a central nervous system is. So you, for example, saw the stimulus, a glass of water in front of you. It was received by your eyes. The sensory neuron carried it to your spinal cord and brain. The relay neuron is actually the intermediate pathway for it. And the signals were sent to a motor neuron, which signaled your effector organ, the muscles, to grab the water and drink it. Another example is when you are presented with a threat or a stimulus that can hurt you. So the stimulus here is the cactus shown, which was received by your touch receptors in your hands. The information was sent to the spinal cord and the brain, relayed within the spinal cord and the brain, and the motor neuron carries it to the effector, which allows you to move your hand away from the stimulus, from the danger. So what are these sensory neurons, relay neurons, and motor neurons? They are just a biological name for nerve cells. So every system is made up of organs. Organs are made up of tissues coming together. Tissues are made up of cells. So the cells in the nervous system, they are known as nerve cells or neurons. And how is information in the nerve cells or neurons transmitted? It travels along the neurons in the form of electrical impulses, electrical signals. So this is the breakdown of your nervous system, which includes the central nervous system and the peripheral nervous system. The central nervous system consists of the brain and spinal cord. These are the two organs involved in the system. And the peripheral nervous system includes your cranial nerves and spinal nerves. Right here in this picture, you can see the brain. And in the center, you can see the spinal cord running down from the brain till the end. Now, the nerves you can see coming out of the brain are cranial nerves. And the ones coming out of the spinal cord are the spinal nerves. So since this exists on the peripheries or on the ends of your body, that is why it is known as the peripheral nervous system. And central nervous system, you can see the brain and spinal cord are in the center of your body. So these structures or these neurons or these nerve cells, they have a structure. And the sensory neurons and the motor neurons, they have different structures, which makes them adaptable to their functions. So we have a motor neuron where the cell body of it as one end of the fiber in the central nervous system. And we have the dendrons, which are fine cytoplasmic extensions of the cell body. And the dendrites are fine extensions of dendrons. So this right here, you can see the motor neuron. This is the cell body, which has the nucleus. These dendrons are extensions of the cytoplasm or the cell body, and dendrites are further extensions. This part of the motor neuron lies in the CNS. Another structure that extends out of the cell body is known as the axon, right here, which goes down to the axon terminals on the muscle fibers, neuromuscular junctions. Just a second. So we said that the cell body of it as at one end of the fiber is in the CNS for the motor neuron. We have dendrons, which are fine cytoplasmic extensions of the cell body, and dendrites are finer extensions of dendrons. There can be junctions with other neurons on any part of the cell body, dendrons or dendrites, and these junctions are called synapses, which will be clear to you further in this presentation. One of the extensions from the motor neuron cell body is much longer than the other dendrons. This is the fiber that carries impulses to the effector organ and is called the axon, which I showed you right here, this longer extension. At the end of the axon, furthest from the cell body, it divides into many nerve endings. These fine branches of the axon connect with a muscle at a special sort of synapse called a neuromuscular junction. In this way, impulses are carried from the CNS out to the muscle. The signals from nerve impulses are transmitted across the neuromuscular junction, causing the muscle fibers to contract. The axon is covered by a sheath made of fatty material called myelin, and this sheath insulates the axon, preventing short circuits, like I said, because the information travels as electrical impulses. So this myelin sheath provides the axon with the protection, and it also speeds up the conduction of the impulses. The sheet is formed by membranes of special cells that wrap themselves around the axon as it develops. Again, here, colored yellow, you can see the myelin sheath, the direction of the impulses, and this provides it insulation and protection. And then the other neuron we have is the sensory neuron, which has a similar structure to the motor neuron, but the cell body is located on a side branch of the fiber, just outside of the CNS. So the motor neuron, the cell body was in the CNS. In the sensory neuron, it is outside the CNS. The fiber from the sensory receptor to the cell body is a dendron, while the fiber from the cell body to the CNS is a short axon. So here you can see the sensory neuron, where this is the cell body. Again, you have the axon on this side. You have the junctions with the sensory receptors. This part is in the CNS. The cell body you can see lies outside of the CNS. And sensory neuron is also covered by myelin sheath, again, protecting it and insulating it. So these are the cells of the central nervous system, the neurons. Now, since we know their structure, we'll see how they relay the information as electrical impulses. So they have synapses, which are critical to the working of the nervous system. The CNS is made up of billions of nerve cells, and these have links with many other through these synapses. So here you can see that these are the neurons, and connecting them is a synapse. So a synapse is actually a gap or a junction between two nerve cells. This gap is not crossed by electrical impulses passing, but by chemicals. Just a second. So here the electrical impulses, they change into chemicals. The impulses arriving at a synapse cause the ends of the fine branches of the axon to secrete a chemical called a neurotransmitter. Right here, you can see these neurotransmitters. They are carried to these synapses. This chemical diffuses across the gap and attaches to the membrane of the second neuron. So this is the receiving neuron. It then starts up impulses in the second cell. This way, they ensure that impulses travel in one direction only. So the neurotransmitter is released by only the presynaptic cell, and it is received by the postsynaptic cell. After the neurotransmitter has passed on the message, it is broken down by an enzyme. And these are specific. So you can see that the chemicals released, they go on to bind with their specific receptors. So these are specific to their receptors, the chemicals. So one of our major and most important sensory receptors is the human eye, which we'll study. Otherwise, our receptors, and they're constantly working, are eyes, as I just said, ears, tongue, nose, and the skin. Important for sight, hearing, and balance, taste, smell, and touch, respectively. So studying the eye, we can see that it can detect light, form an image, and distinguish colors. So it is doing this at the same time simultaneously. So it is receiving three types of informations, and it is also helping us carry out the functions accordingly. The structure of the eye, it includes the iris. What we'll do is we'll start with the outermost layer. So we have the sclera, which you can see is the tough outer coat, visible and wide part of the eye. So it goes around the whole eyeball or the whole eye. Then we have the choroid, we have the cornea. So choroid is underneath the sclera. is a dark layer because it contains pigment cells and blood vessels. The pigment stops the light being reflected around inside the eye. The cornea, sclera, becomes a transparent window and lets light into the eye. So this cornea is actually continuous with the sclera. And it becomes a transparent window. We then have the iris, which is behind the cornea and is the colored ring of tissue. And between the iris, we have the pupil in the middle, which is a hole which lets the light pass through to the inside of the eye. We have the suspensory ligaments, which is a series of fibers that hold the lens in place. So behind or inside the pupil, we can see the lens here, which is held by suspensory ligaments right here, these thin red string-like structures. And then we have the ciliary muscles, the ring of muscles to which the suspensory ligaments attach. The light enters, hits the lens, and is directed towards the retina. The retina is the innermost layer of the back of the eye. It is a light-sensitive layer, and here the light energy is converted into electrical energy of nerve impulses. So this is where our image is formed, or where the light is directed. One part on this retina is the foci, which are cones, are particularly concentrated at the center of the retina in this area. We'll study what cones and rods are. And then we have the optic nerve, which is the sensory neurons. This is the pathway along which the sensory neurons pass and transfer the information to the brain, and the brain then forms the image for us. Now, along this retina, you can see there's a blind spot. So if the light falls on this spot, this is where we cannot see anything, which is why it is known as the blind spot. So how is the eye coordinating, or how is our nervous system coordinating all these three functions? So we have the iris reflex, which is to control the amount of light entering the eye by changing the size of the pupil. We have the circular muscles shown right here on the image, form a ring shape in the iris. And then we have the radial muscles shown right here. These dark red lines are the radial muscles, which are like the spokes of a wheel. So in bright light, our pupil constricts or becomes smaller because the circular muscles contract and the radial muscles relax. We need to make the pupil smaller so that a lot of light does not enter and it does not damage our retina. In dim light, the opposite happens, where the pupil has to dilate to let enough light enter so it can hit the lens and the light can be directed onto the retina, and so we can see and the signals are sent to the brain and it forms an image for us. So in dim light, the radial muscles contract and the circular muscles relax. So the opposite of bright light is happening in dim light. How does it then form an image? To form an image, on the retina, light needs to be bent or refracted. Refraction takes place when light passes from one medium to another of a different density. So in the eye, this happens first at the air or cornea boundary, right here. And again at the lens. In fact, the cornea acts as the first lens of the eye. And as a result of refraction at the cornea and lens, the image on the retina is inverted and upside down. So because the light bends, the image form is inverted, which is upside down. The brain interprets this image the right way up. So it is very necessary for the information to pass through the brain, which is done by sensory, relay, and motor neurons. Then we have another function which helps us distinguish colors. So the retina contains receptor cells called rods and cones, which react to light, producing impulses in sensory neurons. The sensory neurons then pass the impulses to the brain through the optic nerve. The rods, they work in dim light. They cannot distinguish between colors, so brain sees an image produced in black and white. And in dim light, only these rods are working properly. Cones work in bright light. There are three types which respond to different wavelengths or colors of light, which is red, green, and blue. If red, green, and blue are stimulated equally, we see white, and it gives us a sharper image. Both rods and cones are found throughout the retina. Cones are particularly concentrated at the fovea, which we did right here. So this region has a lot of cones, and they're particularly concentrated at the fovea to give us a colored or a white image, give us a sharper image. And then there's a fourth function, which is accommodation, with changes that take place in the eye, which allow us to see objects at different distances. So objects that are near to us and objects that are far from us. This is done by accommodation. How is it done? The shape of the lens can be changed. A lens that is fatter in the middle, which makes it more convex, will refract light rays more than a thinner lens, which is a convex lens. The lens in the eye can change shape because it is made up of cells containing an elastic crystalline protein. So when we have to focus on a distant object, the lens is less convex. Right? These ciliary muscles relax, suspensory ligaments are pulled tight, and the lens is flat. And when we have to focus on a nearby object, the lens is more convex, which is rounded. So the ciliary muscles contract, suspensory ligaments slack, and the lens is more rounded. Okay, now I said there are two systems that are included under the nervous system or needed for coordination and response. And the second one is the endocrine system. So here, a gland is an organ that releases or secretes a substance. So this is happening within our body. We don't see any external or anything externally happening. This is all controlled within our body. The substances released, they also send signals to the brain, and they make changes within the body. So I don't see anyone waiting in the room. So there are two types of glands. We have exocrine glands and endocrine glands. They secrete their products through a tube called a duct. For example, we have the salivary glands in the mouth, which secrete saliva down the salivary ducts, and tear glands, which secrete tears through ducts that lead to the surface of the eye. And then we have endocrine glands, which have no duct, and they are called ductless glands. Instead, their products, which are the hormones, they're secreted into the blood vessels that pass through the gland. Now, hormones only affect particular tissues or organs if the cells of that tissue or organ have special chemical receptors for the particular hormone. For example, the hormone insulin affects the cells of the livers, which have insulin receptors. So the hormone will only go and affect the organ that can receive those chemicals, or those that have receptors for that particular hormone or that chemical. So these are the major endocrine glands that are shown here, which includes the pituitary gland, thyroid gland, the pancreas, adrenal glands located at the top of our kidneys, the testes, and the ovaries. These are the hormones that they secrete, and these are some of their functions. So the pituitary gland, it secretes the follicle stimulating hormone, luteinizing hormone, and antidiuretic hormone. Just give me a second. Let me let in the other participants. Could one of you please unmute your mic and let me know if you can hear me or if you can see the presentation? Anyone, just volunteer and let me know if you can hear me and see the presentation.
We can hear you, but we can't see the presentation.
You can see my screen though, right?
Unfortunately not, miss.
How about now? Just give it a minute.
Yeah, we can
You
now.
can see it now? Is this from the start or this is just, it just happened that you couldn't see the screen? Hello?
Yes, ma'am, I can see it.
Okay, good. Okay, so I said the pituitary gland, it secretes these three hormones mentioned: follicle stimulating hormone, luteinizing hormone, antidiuretic hormone. FSH, it stimulates egg development and estrogen secretion in females and sperm production in males. LH stimulates egg release, or known as ovulation in females, and testosterone production in males. And ADH controls water content of the blood. Thyroid hormones, thyroid gland secretes thyroxine, which controls the body's metabolic rate. Pancreas, they secrete insulin and glucagon. Insulin lowers blood glucose and glucagon raises blood glucose. The adrenal glands secrete adrenaline, which prepares the body for physical activity. These include fight, flight, and fright. Testes secrete testosterone, which controls the development of male secondary sexual characteristics, and ovaries secrete estrogen and progesterone. Estrogen controls the development of female secondary sexual characteristics, and progesterone regulates the menstrual cycle. So one of the few hormones that are important is the ADH, antidiuretic hormone. Sorry, the adrenaline, which is secreted by the adrenal glands. As I said, it prepares you for fight, fright, or flight. So fight, you get ready for fighting, as the name says. Fright, either you stand still and you stop responding, or flight, you run away. So what is happening is the breathing rate increases. Your breathing becomes deeper. You take in more oxygen into the body. Why? Because the muscles, they start to work faster and they get ready. So they need a greater oxygen supply to produce those responses. The heart beats faster, sending more blood to the muscles so that they receive more glucose and oxygen for respiration. Blood is diverted away from the intestine and into the muscles. Why is this happening? Because you do not need to carry out digestion when you are in a fight or flight mode. In the liver, your stored carbohydrate, which is glycogen, is changed into glucose and released into the blood because, as the first point says, the point before, you need more glucose. You need more energy for that respiration to occur and produce more oxygen. So in the liver, the stored carbohydrate glycogen is changed into glucose and released into the blood. The muscle cells absorb more glucose and use it for respiration. Your pupils dilate, increasing your visual sensitivity to movement. Body hair stands upright, making the animal look larger to an enemy. Your mental awareness is increased, so reactions are faster. So your reflexes are faster, and you can give a faster reaction. And there's increased, it is also increased during a race. For example, you have an exam or a job interview, which we call anxiety. So another hormone we said is the insulin, which is released by the pancreas, which lowers your blood glucose level. Again, is made by special cells in the pancreas. These, just for your information, are known as beta cells of the pancreas. It stimulates the liver cells to take up glucose, convert it into glycogen, so that there are lower levels of glucose in the blood. The concentration of glucose in your blood will start to rise after you have had a meal. Sugars from digested carbohydrate pass into the blood and are carried to the liver in the hepatic portal vein. In the liver, the glucose is converted to glycogen, so the blood leaving the liver in the hepatic vein has a lower concentration of glucose than when it enters the liver. Now, if this is disrupted or not happening for some reason, we call it as diabetes. The pancreas cannot make enough insulin to keep their blood glucose level constant. It rises to a very high concentration, which is known as diabetes. So the symptoms, or how can you recognize you have diabetes, is glucose is excreted in the urine due to high concentration of glucose in the blood. So the kidneys start to filter out excess glucose. You constantly feel thirsty, and severe diabetes is a very serious condition. If untreated, it leads to weight loss, and the peak eventually falls into a coma and dies. The treatment for it is you limit the amount of carbohydrate that you eat, because carbohydrates, they contain the most amount of sugars, which is glucose, which gives you instant energy and it increases your blood sugar levels instantly. Or you can have daily injections of insulin. So you are actually consuming the hormone externally from other sources other than your pancreas to keep the glucose in the blood at the right levels. So the differences between coordination and response systems, which are the nervous and the endocrine systems, are the nervous system, they work by nerve impulses transmitted through nerve cells or neurons. But there are chemicals that are released at the synapses. The endocrine system, on the other hand, it works by hormones transmitted through the bloodstream. You have nerve impulses which travel faster and usually have an instant effect, whereas in the endocrine system, the hormones travel slowly and generally take longer to act. An exception to this could be insulin and glucagon, which acts faster, but other hormones like the thyroid glands releasing thyroxine, let's say the testosterone, estrogen, progesterone, they all have a slower effect or slower action. In your nervous system, your response is usually short-lived. So when you're done with your reception and action, the nervous system response is short-lived. Whereas the endocrine system, your response is usually longer lasting. Impulses in the nervous system, they act on individual cells such as muscle fibers, so they have a very localized effect, and hormones can have a widespread effect on different organs, even though they act on particular tissues or organs if the cells have the correct receptors. These were two systems that we covered in coordination and response. Other systems which are involved in our homeostasis and excretion are the urinary systems, integumentary system, which includes our skin, and the respiratory system. So homeostasis is actually the maintenance of our internal environment. So how are we always kept at a constant or steady internal environment, or our organs or parts of our body, is through homeostasis. And excretion generally is getting rid of waste products from our body. So starting with homeostasis, it is the maintenance of a constant internal environment. The inside of the body is known as your internal environment. You have heard of environment, which means surrounding of an organism. The internal environment is the surroundings of the cells inside the body, which particularly means the blood together with another liquid called a tissue fluid. Again, we study this in detail when we cover the circulatory system. The tissue fluid is a watery solution of salts, glucose, and other solutes. It surrounds all the cells of the body, forming a pathway for the transfer of nutrients between blood and the cells. Tissue fluid is formed by leakage from blood capillaries, and it is similar in composition to blood plasma, but just lacks the plasma proteins. So this is just an introduction of what a tissue fluid is inside our bodies and how, so that we can study how it is involved in constantly maintaining our internal environment. So homeostasis includes drinking excessive water, producing more urine. So you drink more water, obviously the water content in your body increases. So as a response, or to maintain the internal environment, you produce more urine. Another example would be increase in body temperature, producing more sweat, and excess proteins, which are excreted as nitrogenous waste. So excretion is also helping out with stasis. Now, excretion is the removal of waste products, mainly urea and ammonia, carbon dioxide, and salts. When the body gets rid of solid waste from the digestive system in the form of feces, this is not excretion. It contains few products of metabolism, just the remains of undigested food, along with bacteria and dead cells. So there's a major difference between excretion and this process right here, when the body gets rid of solid waste product, is known as egestion. We have these four organs that are performing excretion. The lungs get rid of carbon dioxide, which is a waste product, metabolic waste product. The kidneys, again, getting rid of urea and other nitrogenous waste products. The skin gets rid of water, salts, and urea. But these are incidental losses, and small amounts of these products are excreted. And the liver, which carries out deamination to form the urea. So our first system, the urinary system, and how it is involved in excretion. This diagram right here is showing all the organs that are involved, are part of the urinary system. So first, we have the kidneys right here, the main organs of the urinary system. We have from the kidneys, the ureter, which leads to the urinary bladder, and then it leads to urethra to outside of our body. And then we have the blood supply to the kidneys, and this all makes the urinary system. So the urine leaves the kidneys through two tubes called the ureters right here. Urine is stored in a muscular bag called the bladder right here. The wall of the urethra contains two ring-shaped muscles called sphincter muscles. They can control the release of urine, how they can contract to close the urethra and hold back the urine. The lower sphincter muscle is under conscious control or voluntary control, which means it is under are control, while the upper one is involuntary. It automatically relaxes when the bladder is full. So this is the pathway of the urine from the kidneys to outside of our body. The blood supply, each kidney is supplied with blood through a short renal artery. This leads straight from the body's main artery, the aorta, which is right outside the heart. So the blood entering the kidney is at a high pressure. Inside each kidney, the blood is filtered, and the clean blood passes out through each renal vein to the main vein or vena cava. The urethra is just the bladder has a tube leading to the outside called the urethra. Now, how is the excretion carried out by the kidneys? We will study that in more detail. For that, we will have to study the structure of kidneys. This is what the insides of our kidneys look like. So they've sliced it longitudinally, and we can see the inside of the kidneys right here. The darker outer region is called the cortex. This contains tiny blood vessels that branch from the renal artery. It contains microscopic tubes that are not blood vessels. They are filtering units called kidney tubules or nephrons. The tubules then run down through the middle layer of the kidney, which is right here, called the medulla. The medulla has bulges called pyramids, if you can see right here. So these are shaped like pyramids, triangular shape, pointing inwards towards the concave side of the kidney. The tubules in the medulla eventually join up and lead to the tips of these pyramids, where they empty into a funnel-like structure called the pelvis. The pelvis connects with the ureter, carrying the urine to the bladder. So this right here, this white thread-like structure you can see is known as the nephron, which is the filtering unit of the kidney. So it goes from the cortex into the medulla and then to this pelvis area. This unit, filtering unit, is actually performing filtration of all the substances that lead to the kidneys and then which are filtered out and excreted outside our body. How does this nephron work? Let's study that. So this is the structure of the nephron. It is magnified, so we can see it clearly. At the start of the nephron is a hollow cup of cells called the Bowman's capsule. Right here, this cup-shaped structure at the start of the nephron is known as the Bowman's capsule. It surrounds a ball of blood capillaries, which is known as glomerulus. It is here that the blood is filtered. So obviously the blood coming into this glomerulus is bringing all the nutrients and all the waste products from all around the body right here. So the blood is filtered here, and so the exchange of materials takes place here in the Bowman's capsule. And then whatever filtration is done and whatever materials are left in the blood, they are carried out through to the rest of the body. So here in the cortex, you can find the Bowman's capsule, which leads then down to the medulla right here, known as the loop of Henle, coming back to the cortex with second coil tubule, and then right here, this structure is known as the collecting tubule. All of the other tubules of all the nephrons join here so that they can be filtered. And this is the opening at the tip of the pyramid, which leads to the pelvis area, and then into the ureter, then into the bladder. So let's see what products are filtered out of the blood and then back into the blood. So blood in the glomerulus and the space in the capsule are separated by two layers. Right here you can see at the right side of right corner. The capillary wall and the wall of the capsule. So these are cells of the capillary wall and the cell of the Bowman's capsule. Between the two layers is a third layer called the basement membrane. Right here, which is yellow colored, is known as the basement membrane, which is not made up of cells. So these layers act like a filter. They allow water ions and small molecules like glucose and urea to pass through, but they hold back blood cells. So you can see right here, the red blood cells cannot pass through. And large molecules such as proteins, they cannot pass through. So the fluid that enters the capsule space is called the glomerular filtrate. This process where the filter separates different size molecules under pressure is called ultrafiltration. So you can see blood enters here. You can see filtrate passes through into the capsule. This is the layers and the wall shown, and the start of the tubule shown. So this is how ultrafiltration is being carried out here, by stopping some of the materials to not pass through and allowing other materials to pass through. Again, what materials can and cannot pass through, we'll study that here. So the flow rate is actually important here, which is a measure of how much water is in the tubule. If the flow rate falls from 100% to 50%, this is because 50% of the water in the tubule has passed back into the blood. So water does come out and then it passes back into the blood. So sample 1 here, it shows you the blood plasma right here, where the blood is coming from the renal artery. And these proteins, all of these materials are represented by these letters. So protein is represented by A, glucose by B, urea by C, and sodium ions by D. So sample 1 is the blood plasma, which carries all of these materials, protein, glucose, urea, and sodium ions. Sample 2, the glomerular filtrate flow rate is 100%, which means that the water in the tubule has not passed back yet. So in the blood, where sample 1, the plasma contains many dissolved solutes, including protein, glucose, urea, and salts. Protein molecules are too big to pass through into the tubule. So protein molecules, they do not come out of the blood. They stay in the blood. The other substances are at the same concentration as in the blood. So glucose is B, urea is still C, and sodium ions are still D. In sample 3, when the filtrate reaches here, the end of the first coiled tubule, the flow rate is 20%, which means what? 80% of the water has been reabsorbed into the blood. Sodium, 80% of this is reabsorbed, and some of the urea too. Glucose concentration is 0 here. So all of it has been reabsorbed into the blood. Now, when this passes down the loop of Henle here and then enters the collecting duct, the flow rate here is 1, which means that water, 99% of water has been reabsorbed. It has been passed back into the blood. Sodium, urea, protein, and glucose are 0. So all of them have been reabsorbed. Protein never left the blood, right? It never filtered out into these tubules. So it is still present. It is still 0 in this filtrate, and it still exists. All of it still exists in the blood. It was too big to pass through. Urea is still in the fluid. Sodium is only 2D. Not all has been reabsorbed. Some of it still remains in this tubule or in this collecting duct, but is still twice as concentrated as in the blood. So there is more sodium ions in this collecting duct than the blood or the original sample compared to sample 1. So these are all the substances that remain in sample 4. They are then filtered out. So it is mainly urea and sodium ions. Now here you saw that all of the water had been reabsorbed back into the blood, which is why we say that kidneys also control body's water content. So kidneys can control the concentration of the urine and so regulate water content of the blood. If there is increased water intake, more volume of dilute urine will be found or will exist. These changes are controlled by the antidiuretic hormone, which is produced by the pituitary gland. How is this done? So, for example, there is heavy sweating and it is not replaced by drinking. The water content has not been replaced by drinking. Body loses too much water. The antidiuretic hormone starts to work. Concentration of the blood starts to increase. This is detected by receptor cells in a region of the brain, which is known as the hypothalamus, found above the pituitary gland. It causes the pituitary gland to release more ADH, more antidiuretic hormone. This hormone travels in the bloodstream to the kidneys. It causes the collecting ducts to become more permeable to water. So here, the collecting ducts, they become more permeable to water. So any water that was found in the collecting ducts is reabsorbed back into the blood, which makes the urine more concentrated. So when there is less water in the collecting ducts, the urine becomes more concentrated, body loses less water, and blood becomes more dilute. If there is too much water in the blood, for example, we've been drinking too much water, the opposite of all this happens. And so you will find more water in the collecting ducts, which is then passed out of the body through our ureters into the bladder and through the urethra outside of our body. This is the principle of negative feedback, where a change in conditions in the body is detected and starts a process that works to return conditions to normal. When the conditions are returned to normal, the corrective process is switched off. This is part of homeostasis. So any change in the environment leads to correcting that change. It involves the principle of negative feedback. The second system which is involved in homeostasis is the integumentary system, which includes our skin. Skin is the largest organ of our body, and the human skin has a number of functions related to the fact that it forms the outer surface of the body, which include what? Forming a tough outer layer, which is able to resist mechanical damage. It acts as a barrier to the entry of pathogens. It forms an impermeable surface, preventing loss of water, acting as a sense organ for touch and temperature changes, and controlling the loss of heat through the body surface. Okay. Before going into the details of this system, I will have to take a 10-minute break to offer my prayers. So everybody stay in the meeting. Just keep your mics on mute, take a break, and come back in 10 minutes. And now you can again see my screen. Okay, coming back to the integumentary system, which is another system that is involved with homeostasis and excretion. We've already discussed these functions of the human skin. It resists mechanical damage. It is a barrier to entry of pathogens. It prevents loss of water, is a sense organ for touch and temperature changes, and controls the loss of heat through the body surface. These are the layers of the skin. These are the parts that are forming our integumentary system. So we have the epidermis, which you can see is the top layer of our skin, which consists of pores and the hair. The outer layer consists of dead cells, stops water loss, and protects the body against invasion by microorganisms such as bacteria. Underneath the epidermis, we have the dermis, which is the middle layer, and it consists of the most important parts of the skin. It contains sensory receptors, is the location of sweat glands, has many small blood vessels and hair follicles. The last three structures are involved in temperature control. So, sensory receptors, sweat glands, and blood vessels and hair follicles, these are involved in temperature control. We have the hypodermis, which contains fatty tissue. It insulates the body against heat loss and is a store of energy. The sweat glands, which you can see right here, produce greater amounts of sweat and which is secreted onto the surface of the skin through here. So you can see it comes out of the pore. Now, how does this control our body temperature? In humans and other mammals, the core body temperature is monitored by the part of the brain called the thermoregulatory center. Again, located in the hypothalamus of the brain, it acts as the body's thermostat. The hypothalamus detects a rise in the body temperature, core body temperature. Immediately, it sends nerve impulses or electrical signals to the skin, which brings about the following changes to correct the rise in Okay, coming back. So the following changes happen to correct the rise in temperature. The sweat glands, they produce greater amounts of sweat. This liquid is secreted onto the surface of the skin. When a liquid evaporates, it turns into gas. When sweat evaporates, it cools the body down. So it is not actually the sweat which is cool. It is secreted at body temperature. It only has a cooling effect when it evaporates. So in very humid atmospheres, example, a tropical rainforest, the sweat stays on the skin and does not evaporate. It then has a very little cooling effect. Okay, is anyone saying something? If it's a question, you can keep it for the end of the session. Okay, I guess not. So then come the hairs. Hair on the surface of the skin, they lie flat against the skin's surface. This happens because of the relaxation of tiny muscles called hair erector muscles, which are attached to the base of each hair. In cold conditions, these contract and the hair are pulled upright. The hair trap a layer of air next to the skin, and since air is a poor conductor of heat, this acts as insulation. What happens in warm conditions is the thinner layer of trapped air means that more heat will be lost. This is not very effective in humans because the hair over most of our body, they do not grow very large. It is effective in hairy mammals. For example, we have dogs or cats. And the same principle is used by birds, which fluff out their feathers in cold weather. The third are the tiny blood vessels called capillary loops in the dermis. The blood flows through these loops, which radiate the heat to the outside, and they cool the body down. If the body is too hot, the arterioles, which are small arteries, which lead to the capillary loops, they dilate. When they dilate, this brings in more blood, so increasing the blood flow to the skin surface, which is called vasodilation. So vaso means vessel, dilation means increasing or dilating. In cold conditions, the opposite happens. The arterioles leading to the surface capillary loops, they constrict. When they constrict or become narrow, the blood flow is reduced. So there is no heat loss. This is called vasoconstriction. So again, all of these parts or all of these organs of the integumentary system, they are acting to maintain homeostasis, to maintain a constant internal environment. Then comes the role of liver in excretion. It basically just breaks down excess amino acids and produces urea. Breaking down of amino acids is known as deamination. So we are removing the nitrogen-containing part of amino acids to form the urea. Urea is actually the excretory product, which is excreted through kidneys. Then we have the yellow-green bile pigment, bilirubin, which is a breakdown product of hemoglobin. The bilirubin is excreted with the bile into the small intestine and expelled with the feces. It basically undergoes changes in the intestine and is largely responsible for the brown color of our feces. Next, we have breathing and respiration, which is done by our respiratory or gas exchange system. Breathing and respiration are two separate processes, but basically our gas exchange system is also known as our respiratory system. So what are these two processes? Cells get their energy by oxidizing foods such as glucose during the process called respiration. If the cells are to respire aerobically, they need a continuous supply of oxygen from the blood. Carbon dioxide from respiration needs to be removed from the body. So in humans, these gases are exchanged between the blood and the air in the lungs. So breathing is what supplies us with the oxygen from the external environment or the air outside of the body and into our bodies. And carbon dioxide we get rid of. So this is happening via gas exchange in our lungs. Respiration, on the other hand, is the oxidation reaction that releases energy from foods such as glucose. So again, what is the difference? Respiration is the oxidation reaction, releasing energy from foods. Breathing is the mechanism that moves air into and out of the lungs, allowing gas exchange to take place. The lungs and associated structures are often called the respiratory system, but this can be confusing because respiration is a separate process. And so it's better that we call them a gas exchange system. This is our gas exchange system. So these are the lungs, the main organ of the gas exchange system, and its associated structures. Here you can see we breathe in through the nose or the mouth, and it travels down the trachea into the lungs. Now, the lungs, they are enclosed in the chest or the thorax by the rib cage and a muscular sheet of tissue called the diaphragm. So the boundaries of our lungs are the chest or the thorax, and here at the bottom you can see this curved muscle is the diaphragm. Joining to each rib to the next are two sets of muscles called intercostal muscles. Costals means rib and inter means in between. So in between each rib, you can see these red structures, which are the intercostal muscles. The diaphragm here separates the contents of the thorax from the abdomen. So this is also a separating boundary between the chest and the abdomen. Diaphragm, you can see, is dome-shaped. It has a fibrous middle part. So the fibrous part of the diaphragm is right here, and the muscular part of the diaphragm is right here. We have the trachea, or the walls of the trachea and bronchi. So here we have the trachea, contain rings of cartilages, and it travels down to the bronchi. You can see the structure right here. So these are the rings of cartilage in between. Now, the inside of the thorax is separated from the lungs by two thin, moist membranes called the pleural membranes. Right here, if you can see, is the pleural membrane. One of them is lining on the outside near the rib cage, or the ribs and the intercostal muscles, and one of them is lining the lungs. So the inside of the thorax is separated from the lungs by these two membranes. They make up a continuous envelope around the lungs, forming an airtight seal. Because if there was not an airtight seal, air would easily enter and exit through here, and we would not need this whole system. Between the two membranes, you can see the blue part is a liquid. There's a pleural cavity, which is filled with pleural fluid. Anything that has the word pleural in it is related to the lungs, and also whenever you hear the word pulmonary, it is related to the lungs. Now, this pleural fluid, it acts as lubrication so that the surfaces of the lungs, they don't stick to the inside of the chest wall when we breathe. So this allows reduced friction and easy gliding of all these structures inside the pleural cavity to make breathing easier for us. Now you can see the trachea and the rings of cartilage, they go down to the bronchi. These are the two bronchi. Then we have the bronchioles. So the bronchi divide into bronchioles, and at the end of the bronchioles, you can see alveoli. So these little cloud-like structures, these are the alveoli. These are microscopic air sacs. This is where gas exchange takes place between the alveoli and the blood. How is this happening? Again, let's study that. Now, first, there are some protective functions of the trachea or the parts of the gas exchange system. The trachea and larger airways are lined with a layer of cells that have an important role in keeping the airways clean. So basically, right here, you can see the trachea. These are lined by some protective cells, which are goblet cells, which secrete a liquid called the mucus. The mucus traps particles of dirt or bacteria that we breathe in through our nose and mouth. Then we have ciliated cells, which are covered with tiny hair-like structures called the cilia, hence ciliated cells. These cilia, they beat backwards and forwards, which can sweep the mucus and the trapped particles out towards the mouth. Which is why whenever any foreign bacteria enters, we cough it out. And so dirt and bacteria are prevented from entering the lungs. And so we can avoid an infection. Now, we said that gas exchange takes place. But before that, we need to study what ventilation is. Ventilation means moving air into and out of the lungs. This requires two very important things, which is a difference in. So the air moves from a place where the pressure is high to one where it is low. And it also depends on the fact that thorax is an airtight cavity. Like I said, if this wasn't an airtight cavity, we would just have a gas exchange system occurring right here, right next to the lungs. When we breathe, we change the volume of our thorax, which alters the pressure inside it, and this causes air to move in or out of the lungs. There are two movements that bring about ventilation. Those are movements of the ribs and the diaphragm. Again, when we're breathing in, we're increasing the volume of our thorax, which brings in air. And when we breathe out, we squeeze out the air. We decrease the volume of our thorax, and then we breathe out. So this is how pressure changes takes place, which allows us to breathe. This is what is happening. So you can see right here, the top right corner shows you the rib cage and the intercostal muscles. We have two types. One of them on the inside of our ribs, which are known as internal intercostals, and the external intercostals found on the outside of our ribs. And this is the sternum, the middle bone that runs down between our ribs. And this is the sliced version, you can say. So these white round structures, these are the ribs, and in between we have the intercostal muscles running. So during inhalation, what is happening is when we breathe in, the diaphragm muscle is originally a dome-shaped muscle. When we breathe in, it flattens. And so it increases the thoracic cavity. Air enters our lungs, and since we have lower pressure than the outside, it is easier for air to move from a higher concentration to a lower concentration. Again, we can breathe in enough air because the diaphragm has become flat and it has increased the volume of our thorax. The external intercostals, the external intercostal muscles, they contract, pulling the ribs up and out, again increasing our thoracic cavity. When we exhale, the air is being pushed out because there's a higher pressure in the lungs than the outside of the body. Again, diaphragm snaps back, not snaps back, but it comes back to its original relaxed position, which is the dome-shaped position. Becomes curved again. The internal intercostal muscles, they contract. They pull the ribs down and inside. So this is how ventilation is happening inside our lungs. An easier way to remember this is during inhalation, you can remember the mnemonic RICE, where we relax our internal intercostals and we contract our external intercostals. And then during exhalation, you can remember it by airing. So externals relax and internals, they contract. So to remember the movements of the intercostal muscles, you can use this mnemonic. So once the air has entered our lungs, how does it exchange the gases, that is oxygen and carbon dioxide? Now you can see here, this is a magnified structure of the alveoli, which was shown right here. This is the main site of gas exchange. So here in the alveoli, the air enters and leaves. Now you can see that it is covered by a blood supply. It has a blood supply which brings in oxygen, and then it can also exchange carbon dioxide. Blood from the heart enters here and then leaves. It goes back to the heart. Now you can see that there are enormous numbers of alveoli. It has been calculated that the two lungs contain about this much of tiny air sacs, giving it a total surface area of 60 meters square. Again, surface area is too large for efficient gas exchange to take place, and they're covered with tiny blood capillaries, which allows, again, efficient air exchange to take place. Then, when you can see, again, the magnified structure of the blood and the relationship between the blood capillaries and the alveoli, you can see that around the lungs, the blood is separated from the air inside each alveolus by only 2 cell layers, which decreases the distance for the gas exchange to occur. So the shorter the distance, the easier gas exchange takes place. The cells making up the wall of alveolus and the capillary wall itself. So this is the capillary, these are the capillary wall cells, and these are the cells of the alveolus. This distance is less than 1,000 of a millimeter, again allowing easy gas exchange to take place. The blood brings in oxygen. Since this blood carries more oxygen concentration, the air we breathe in carries more oxygen, which means that here the oxygen concentration is higher than in the blood. Why? Because the blood is collecting carbon dioxide that was released during all the respiring cells. So again, a concentration gradient exists. So carbon dioxide moves from a higher concentration that is in the blood to a lower concentration that is in the alveoli. And since the alveoli have a higher concentration of oxygen, it diffuses into the blood capillaries. And this is how gas exchange takes place. Then the blood carries the oxygen to other parts of the body and to the heart. So a concentration gradient has to exist. There is greater oxygen concentration in the alveolus from the air. So oxygen diffuses into the blood, and there's a greater carbon dioxide concentration in the blood. So carbon dioxide diffuses out of the blood into the alveoli or alveolus and is then breathed out. Again, these are all the adaptations of the alveoli to allow efficient gas exchange. It has a very, very large surface area. It has blood capillaries, tiny blood capillaries, a good blood supply. There is a concentration gradient that exists. The distance between the blood capillaries and the alveolus is 1,000 of a millimeter, decreasing the diffusion distance and again, making gas exchange more efficient. Now there's a relationship between breathing rate and exercise. The increased rate and depth of breathing during exercise allows more oxygen to dissolve in blood and supply the active muscles. The extra carbon dioxide that the muscles put into the blood is detected by the brain, which instructs the intercostal muscles and diaphragm muscles to contract and relax more rapidly. This increases the breathing rate. So carbon dioxide will be removed faster by faster and deeper breathing. Again, what is happening during exercise? Because we're using too much of our muscle cells are respiring at a greater rate, which is using up more oxygen. So we need a greater supply of oxygen for the muscles to respire aerobically. This is why we start to breathe faster and deeper so that we can get in more oxygen at a faster rate and supply that oxygen to the muscles. And also so we can get rid of the carbon dioxide faster. So cells in the body are constantly respiring even when we are not doing physical work. They produce carbon dioxide, which is expired by the lungs. During exercise, the cells respire more rapidly, producing more carbon dioxide. Now, smoking. In order for the lungs to exchange gases properly, the air passages need to be clear. Like I said, a protective mechanism was performed by the goblet cells and the ciliated cells. So the alveoli also need to be free from dirt, particles, and bacteria. They must have a big surface area as possible in contact with the blood. Now, there is one habit that upsets all these conditions, which is smoking. And how does it do that? It is associated with all of these different diseases, which reduce the efficiency of the lungs and cause damage to them. So one of them is bronchitis. Now, bronchitis, as the name suggests, is related to the structure, the bronchi or bronchioles. Itis means inflammation. So reduced number of cilia. Because of smoking, you have reduced number of cilia. Mucus is not swept away from the lungs. That mucus blocks the air passages, which is made worse by the fact that smoking irritates the lining of the airways. This in turn, the cells start to secrete more mucus. Again, blocking the air passages. The sticky mucus blocking the airways is the source of smokers' cough, which is why we see them coughing up more, vigorously and more frequently. So irritation of the bronchial tree with the infections from the bacteria and the mucus, they cause bronchitis. They cause inflammation of the bronchioles or the bronchi. Bronchitis blocks normal airflow, so the sufferer has difficulty breathing properly. Then we have emphysema, where smoke damages the wall of the alveoli, break down and fuse them together again. So the original walls of the alveoli, they're destroyed, they're broken down, and they fuse together, and they formed enlarged irregular air spaces. This reduces the surface area for gas exchange. It becomes more inefficient, and blood carries less oxygen. In serious cases, it leads to the sufferer being unable to carry out even mild exercise such as walking. There has been no treatment for emphysema as such, and it often leads to a very slow, painful death. And last but not the least, we have lung cancer, where smoke contains strongly addictive drug, nicotine. It contains chemicals such as carbon monoxide, arsenic, ammonia, formaldehyde, cyanide, benzene, and toluene, which are carcinogens. Carcinogens are pathogens that can cause carcinomas or cancer. These are contained in the tar that collects in the smoker's lungs. What happens in cancers is the cells start to mutate, and they start to divide uncontrollably, forming a tumor. So this allows them to divide very rapidly, and so it leads to a tumor. Another effect that smoking has is if you smoke during pregnancy, pregnant women who smoke, they're more likely to give birth to underweight babies. Now, it's a good thing that the gas exchange system and the circulatory system, they come after one another because they're very closely related. So another function that our body is carrying out is it is carrying blood all around our body, and the blood is circulating all around our body, which keeps our cells living and healthy. Which is done by which system? It is done by the circulatory system, also known as the transport system, because blood transports everything all around the body. So blood is pumped around a closed circuit made up of the heart and blood vessels. Why is there a need for the circulatory system? As it travels around the body, it collects materials from some places and unloads them in others. In mammals, what it does is blood transports oxygen from the lungs to all parts of the body. Obviously, every cell in our body needs oxygen to respire. It carries carbon dioxide from all parts of the body to the lungs. Again, when the cells respire, they give out carbon dioxide, which needs to be excreted out of our body, which is done by the lungs. The blood transports nutrients from the guts to all parts of the body. Again, we need healthy living tissues, so we need cells, so we need nutrients. We need to get rid of the urea from the liver. So it's carried from the liver to the kidneys. We need hormones circulating our bodies. We need antibodies to fight off diseases, to always be circulating in our bodies, and other substances. And it also distributes heat around the body, which we did was known as vasodilation and vasoconstriction. So here you can see that there are two main types of circulatory system in animals. One of them is a single circulatory system where the blood is pumped from the heart to the gas exchange organ and then directly to the rest of the body. This you can see right here. If you can see the fish and the circulatory system inside the fish, this is a single circulatory system. How is the gas exchange or how is the oxygenation taking place of the blood? This is taking place directly. So if you notice the gills on the fish, that is where the gas exchange in the fish takes place. And blood only goes around once in the body through the heart. So which is why it is known as a single circulatory system. Then we have a double circulatory system shown here below the fish, where the blood is pumped from the heart to the gas exchange organ, from the heart to the lungs, and back to the heart, and then to the rest of the body. So it is going through the heart twice, which is why it is known as a double circulatory system. It is more efficient, can handle higher pressures, can maintain higher pressures, and blood also moves very quickly. So in the humans, we have a closed double circulatory system. In the fish, there is an open single circulatory system. Open because gas exchange takes place directly between the air and the blood. In the humans, it takes place between the lungs and outside of our body, and then that is transferred to the heart so that the oxygen from the lungs can be taken all around the body. So we have a closed double circulatory system. This system includes the organs, heart, the main pump of the system, blood vessels, where the blood is carried in or through, and the blood itself, which is the transport medium. Now, the heart, which it says is the pump. The blood vessels, these carry the blood around the body. Arteries carry blood away from the heart towards other organs, whereas the veins carry blood towards the heart and away from the organs. And there is a third blood vessel known as the capillaries, which carry blood through the organs and also link the arteries and the veins. So our closed double circulatory system contains further two circulations, which or two parts to the double circulatory system, which are known as pulmonary circulation and systemic circulation. Pulmonary circulation, as the name suggests, is through the lungs. So deoxygenated blood leaves the heart through the pulmonary arteries and is circulated through the lungs, where it is oxygenated, and the oxygenated blood returns through the heart through the pulmonary veins. So if you come to the diagram, you can see that the blood here leaves the heart, goes into the lungs. This is where the gas exchange takes place, where the blood gets oxygen and gets rid of the carbon dioxide. The lungs then the blood then leaves the lungs through the pulmonary vein. The oxygenated blood then is carried back again to the heart. The heart then takes this or throws this oxygenated blood through here, through the pulmonary vein all around the body, and then oxygenates all the other parts of the body or all the cells takes their carbon dioxide, and then since it is taking in the carbon dioxide, the blood becomes deoxygenated. That deoxygenated blood is returned back to the heart. The heart then sends it back to the lungs to become oxygenated again. So pulmonary circulation involves the exchange between, or the blood circulation between the heart and the lungs. Then we have a systemic circulation. Oxygenated blood leaves the heart through the aorta, right here, is circulated through all other parts of the body where it unloads its oxygen. Deoxygenated blood returns through the heart, through the vena cava. Again, this is the detail of how the heart does this. So here you can see the structure of the heart. The outside shows the right atrium. So there are 4 chambers to the heart. It is divided into right atrium, right ventricle, left atrium, and left ventricle. The main blood vessels of the heart are the aorta, pulmonary artery, pulmonary vein, and the vena cava. The heart itself is supplied by blood as well, because obviously heart needs nutrients and it needs to live to work as a pump. So the coronary arteries, coronary is a word that is related to everything about the heart. So the heart receives its circulation and nutrients through these coronary arteries. And then it supplies the entire system or entire body through these aorta, pulmonary artery, pulmonary vein, and vena cava. When we slice the heart into half in a longitudinal section, this is the inside of the 4 chambers that we can see. So we have the right atrium, the right ventricle, the left atrium, the left ventricle. So what is happening is the atria, they fill with blood. Let's start on the right side. So here you can see the right atrium. The right atrium gets blood from all around the body, right? And it gets deoxygenated blood. So blood that is carrying carbon dioxide. This is brought to the right atrium by the vena cava. So the inferior vena cava brings in blood from regions below the heart, and the superior vena cava brings blood from the regions above the heart, which are the arms, upper arms, and the head and the neck. This deoxygenated blood enters the right atrium, right? Can you see this right atrium filling in? Why is it shown in blue? Because it has the deoxygenated blood. This right atrium then contracts. Can you see it contract? When it contracts, the blood moves from the right atrium to the right ventricle. Now, there are valves that separate the atria from the ventricles. So the right atrium is separated from the right ventricle by the tricuspid valve. This is like a door that can open only in one direction. So you can see right here, the tricuspid valve can only open down, which is why blood from the right atrium enters the ventricles. After it enters the ventricles contract. When the ventricles contract, the blood is pushed to the pulmonary artery, right? Why does it not go to the atrium? Because of these valves. Okay, I think we're back. I hope everyone can hear me again and see my screen again. Okay, where were we? So I said the blood from the vena cava enters the right atrium. When it contracts, it is pushed to the right ventricles. It has to pass through the tricuspid valve. The ventricles, when they contract, they push blood into the pulmonary artery. Backflow of blood from the ventricles into the right atrium is prevented by these tricuspid valves. When the ventricles contract, the blood goes through this valve, which is known as the semilunar valve. Semi means half, lunar means moon shape, and it is a valve. This valve is pushed open and the blood enters the pulmonary artery. Now, there's a contradiction. We said that arteries carry blood away from the heart, which is the pulmonary arteries doing here. But we also said that arteries carry oxygenated blood. Pulmonary arteries are the only exception. They carry deoxygenated blood because they have to carry blood away from the heart into the lungs so that the blood can become oxygenated again. So this blood is carried then to the lungs. In the lungs, the blood is oxygenated because the gas exchange takes place and is returned to the left atrium of the heart by the pulmonary vein. Again, veins bring blood back to the heart and veins carry deoxygenated blood. But the only exception to this is the pulmonary vein, which is bringing the blood back to the heart, but it is carrying oxygenated blood. Why? Because the lungs have oxygenated the blood and the pulmonary veins bring it back to the heart. This blood enters the left atrium through the pulmonary vein. Right? You can see two parts to this pulmonary artery, which is going into each of the lungs. Now, from the lungs, the blood is brought back through the pulmonary vein into the left atrium. The left atrium then contracts, pushing blood into the left ventricle. Again, this blood passes through bicuspid valve, or also known as the mitral valve, which exists between the left atrium and the left ventricle. When the ventricles fill up, they contract, pushing blood into the aorta. The bicuspid valve prevents backflow of blood into the left atrium. The semilunar valves, they open up and they carry blood into the aorta. Now, these valves, semilunar valves in the aorta, also prevent backflow of blood into the left ventricle. This aorta then carries blood to all parts of the body. And this cycle keeps on repeating. Now, if you can see, because there's a difference between the walls of the atria and the ventricles, and there's a difference between the pulmonary arteries and the aorta. So the atria are receiving blood, and the right atrium receives blood, receives deoxygenated blood. It has thinner walls because it does not receive blood with a lot of pressure. Right? And again, the ventricles, because they're receiving blood from the right atrium, the right ventricle receives blood from the right atrium, so it does not have thicker walls. Again, the pulmonary arteries, they don't have thicker walls because they receive blood with less pressure and they only need to carry. And the ventricles have thinner walls as well. Why? Because they only send blood to the lungs or push blood to the lungs, which is very close to the heart. So they don't need that much of pressure, and the pulmonary arteries don't carry that much pressure. The left atrium, again, is similar to the right atrium because it receives blood with low pressure. Then it is the left atrium contracts and pushes blood into the left ventricles. The left ventricles, they have thicker walls. Why? Because they need to push blood with a lot of pressure out into the aorta so that it can be carried all around the body. Now, carrying blood all around the body is a very large distance. So the ventricles have thicker walls so they can push blood with a harder force, with a lot of force. And again, the aorta has thick muscular walls so it can withstand that pressure and again carry blood with high pressure all around the body. Separating the ventricles, there's a septum medium that separates the ventricles and prevents the blood in the ventricles from mixing between each other. I just told you a lot of the structures of the heart that are adapted to its functions. So it is divided into a left and a right side by a wall of muscle called the septum, which is known as the medium septum, which means medium means in the middle, and septum is the wall of muscle that separates them. Right ventricle pumps blood only to the lungs, while the left ventricle pumps blood to all parts of the body. This requires much more pressure, which is why walls of the left ventricles are much thicker than the right ventricles. Valves ensure the blood can flow only in one direction through the heart. Walls of the atria are thin. They can be stretched to receive blood. As it returns to the heart, but can contract with enough force to push blood through the bicuspid and tricuspid valves into the ventricles. Walls of the heart are made up of cardiac muscle. This is a special type of muscle which can contract and then relax continuously without becoming fatigued. Cardiac muscle has its own blood supply, the coronary circulation, which was also shown in this diagram. And blood reaches the muscle via coronary arteries. These carry blood to capillaries that supply the heart muscle with oxygen and nutrients. Blood is returned to the right atrium via the coronary veins. Now, coronary arteries, they're very narrow arteries, and they can be easily blocked by the buildup of fatty substances, which is usually almost always known as cholesterol, which can cut off the blood supply to an area of cardiac muscle. The affected muscle can no longer receive oxygen and glucose. It cannot respire and release energy. This means it is unable to contract, resulting in a heart attack, which is one of the coronary heart diseases that we come across very frequently and commonly. It can lead to severe health problems and is often fatal. Now, the number of factors that contribute to coronary heart diseases are, for example, we have heredity. Some people inherit the tendency to develop these coronary heart diseases. There's high blood pressure, which puts a lot of strain on the heart. Your diet, which might include saturated fats, which raises your cholesterol levels. There's smoking, which increases your blood pressure and makes blood clots more likely to form. There's stress, which also increases blood pressure, and there's lack of exercise. Because exercise helps to reduce blood pressure, and it also makes the heart stronger. Again, the heart is beating at a normal rate, which is known as normal heart rate, which is 70 beats per minute. So in a minute, our heart beats 70 times. When we exercise, the muscles must release more energy. They again require an increased supply of oxygen for aerobic respiration. To deliver that extra oxygen, both the number of beats per minute, that is the heart rate, and the volume of the blood pumped with each beat increases. So the heart rate and the stroke volume both increase to accommodate that requirement when we exercise. Again, when we are angry or afraid, our heart rate again increases. The increased output supplies extra blood to the muscles, enabling them to release extra energy through aerobic respiration. This again allows us to fight or run away and is called fight or flight response. Triggered again by the release of hormones. One of the hormones is adrenaline from the adrenal glands. The ways in which we can monitor the activity of the heart includes pulse rate, measuring the pulse rate, listening to heart sounds, and the use of electrocardiograms. So these are 3 ways we can keep our heart rate in check. Now, this was one organ of this whole circulatory system, which was the heart. The other organs are arteries, veins, and capillaries, the blood vessels that form one of the organs of this system. Arteries again carry blood from the heart to the organs. They carry high blood pressure. They have a narrow lumen and thicker walls. They're very elastic and muscular to withstand the high blood pressure, and they have no valves. Why? Because the pressure and the force of blood pushes it in one direction, pushes the blood in one direction only. So there is no need for valves in arteries. The veins, on the other hand, they return blood to the heart. Blood pressure is steady and low. It has a wider lumen. Why? Because it has thinner walls. It is less elastic and less muscular because it does not need to withstand high blood pressure, and it has valves to prevent backflow of blood. This also ensures that the blood is moving in only one direction. The capillaries, they carry blood through the organs. They allow exchange. So the blood pressure through the capillaries is very, very low. The walls are one cell thick, again, to allow easy diffusion and efficient exchange. They are permeable and they have no valves. These are the structures of each blood vessel shown. So this is the artery, which has thicker walls. It has muscle fibers and elastic tissue. Again, elasticity, why? Because it needs to withstand high blood pressures, and it also experiences pulsations, right? So there is an increase in pressure and then decrease, increase in pressure and then decrease. These are the veins which have thinner walls and a wider lumen. Veins also have semilunar or half-moon shaped valves which prevent the backflow of blood, which is shown here, right? So these veins, once the blood passes through them, it cannot move in reverse. And these are the capillaries which go through each organ, and they allow gas exchange to take place. Anything that will be in a higher concentration will move towards lower concentration. And this is how the capillaries allow exchange of materials between the blood and the cells. The arteries, they divide into arterioles. These arterioles divide into capillaries. Capillaries join back to form venules, and the venules join back to form veins. The third organ that makes up the circulatory system is the blood itself. Why is blood known as an organ? Cells join to make up tissues, tissues join to make up an organ. Now, blood consists of different cells: red blood cells, white blood cells, platelets, plasma, and tissue fluid, which is why it can be called as an organ. And the blood floats around in a liquid called plasma. So plasma is the liquid part of the blood. It is mainly water, carries the blood cells around the body, carries dissolved nutrients, hormones, carbon dioxide, and urea. Also distributes heat around the body. Again, blood is composed of red blood cells, shown right here, white blood cells, and platelets. Each of these have their own functions, and they're adapted to their functions. How? Red blood cells, known as erythrocytes, they're highly specialized cells which are made in the bone marrow. They are destroyed after a lifespan of 100 or 120 days. They're destroyed in the spleen. Their only function is to transport oxygen. So they contain hemoglobin, a special type of protein, an iron-containing protein that combines with oxygen to form oxyhemoglobin. As they pass through the lungs, they load oxygen. As they pass through active tissues, they unload oxygen. Very special thing about the red blood cells is they have no nucleus. This means that more hemoglobin can be packed into each red blood cell, so more oxygen can be transported. They're also biconcave shape. Here you can see their biconcave shape, which allows efficient exchange of oxygen in and out of the cell. This allows them to move in and out of the capillaries, right, without being damaged. They have a high surface area to volume ratio, giving a large area for diffusion. And they are thin shape of the cell. Their thin shape of the cells results in a short diffusion distance to the center of the cell. Platelets are another very important cells of the blood, which are known as thrombocytes as well. They are not whole cells. These are fragments of large cells made in the bone marrow, which, as you can see here, they're little tiny fragments of cells. Basically, they prevent the loss of blood. If the skin is cut, exposure to the air stimulates the platelets right here. So this is the cut that is shown here. This stimulates the platelets and damaged tissue to produce a chemical. This chemical causes the soluble plasma protein fibrinogen to change into insoluble fibers of another protein called fibrin. Fibrin, you can see right here, these are thread-like structures which form a network across the wound. Basically, they form a net across the wound. That net traps red blood cells. It forms a clot, which prevents further loss of blood and entry of bacteria or microorganisms or dirt into our blood system or into our circulatory system. This clot over time develops into a scab, which protects the damaged tissue while new skin grows. The third important cell are the white blood cells, which are also known as leukocytes. There are several types of white blood cells, but we know the two most important are lymphocytes and phagocytes, right? Shown right here. And the main role of white blood cells is to protect the body against invasion by disease-causing microorganisms. So phagocytes, they carry out phagocytosis. These cells are, they form about 70% of the white blood cells, and they can ingest the bacteria. How do they do this? They do this by changing their shape. So there are two extensions of their cytoplasm known as pseudopodia. These pseudopodia surround the bacteria in a vacuole, brings that bacteria inside. The phagocyte secretes enzymes to break down the microorganism, and then it carries out phagocytosis, which means it eats the cell. So here, this diagram shows you each stage of phagocytosis. So these are the extensions, cytoplasmic extensions. Here the bacteria enters. The lysosomes, they cause the bacteria to break down. This is the broken down bacteria, and then it is released. The other very important function is the production of antibodies, which is carried out by lymphocytes, which make up 25% of white blood cells. These antibodies are chemicals which are soluble proteins. They pass into the plasma. Pathogens such as bacteria and viruses have chemical markers on their surfaces, which the antibodies recognize. So these markers are known as antigens. They're basically little markers on the bacteria, which the antibodies recognize, and then they attack those antigens or bacteria. The antibodies stick to the surface antigens, destroy the pathogen. How do they do this? In a number of ways. Causing the bacteria to stick together so that phagocytes can ingest them more easily. They act as a label, which means that they're easily recognized by the phagocyte. They cause bacterial cells to burst open, and they also neutralize poisons produced by the pathogens. Now, this right here, production of antibodies, which follows the first exposure to the entry of a foreign material, is called primary immune response. They form other cells which are known as memory cells. So some lymphocytes, instead of participating in the primary immune response, they develop into memory cells. These cells remain in the blood for many years, sometimes also a lifetime. If the same microorganism enters again, the memory lymphocytes start to reproduce and produce antibodies, so the pathogen can be quickly dealt with. This is known as immunity. So we have developed immunity against the foreign particle. When we develop this immunity, the second immune response that takes place is much faster and more effective than the primary response. Again, a person can be given artificial immunity to a disease-causing organism without actually contracting that disease. So this is done by vaccination. A person is injected with the agent that carries the same antigen. The lymphocytes recognize this. They multiply exactly as if they did when the microorganism entered the bloodstream. They produce memory cells. They make the person immune to the disease. And if the person now comes into contact with the real pathogen, they will experience a secondary immune response. Antibodies will be produced faster in more quantity, and it will be enough to prevent the pathogen reproducing in the body and causing the disease. Now we come to the system that deals with nutrition and digestion, which is the gastrointestinal system. We require nutrition through a balanced diet, which includes carbohydrates, fats, oils. It includes lipids. which is another name for fats and oils, and proteins. These are the sources of these food groups and their uses in the body. The most important source of energy are the carbohydrates, which we get from potatoes, rice, etc. Then there's cheese and milk and butter, which provides us with the lipids, which is another source of energy, deals with insulation or provides insulation. And the cell membrane is also made up of lipids. And then we have proteins, which are important for growth, tissue repair. Enzymes are made up of proteins. Some hormones are made up of proteins. And they are provided, this food group is provided by meat, fish, eggs, etc. Then we have vitamins. The most important ones are vitamin A, vitamin C, vitamin D. Vitamin A is important for our eyes, which prevents night blindness. Vitamin C is important because it acts as a catalyst in cell respiration, and it prevents scurvy. Scurvy is bleeding gums and poor wound healing. So vitamin C is involved in healing process. Vitamin D, again, gives us stronger bones and stronger teeth. Then we have calcium and iron. Calcium, again, makes our teeth and bones stronger. And iron is a very important part of hemoglobin in red blood cells, which helps us carry oxygen. Then we have water, which is a major component of our plasma. And all of these substances, they are carried in water. Body fluids, blood, lymph, and tissue fluid are composed of water. Digested food, and basically it's a carrying medium like blood. Also acts as a solvent, and it is also useful in the process of digestion, which we'll study further. And then fiber, where it prevents constipation, and it also provides bulk and makes us feel fuller. Okay, how does digestion take place in our body? We have the digestive system. These are the organs that make up our digestive system. Please ignore the lungs over here. This is part of the respiratory or the gas exchange system. The alimentary canal, which is the canal that is involved in our digestion, includes the mouth, the salivary glands, esophagus, where the food passes from the mouth into the stomach. We have the duodenum, which is part of our small intestine, the ileum, also part of our small intestine. We have the pancreas, which deal with the glucose and chemical digestion of the glucose that we intake. We have the liver, which again is an important organ for digestion, the gallbladder, the colon, the rectum, and the anus. This is the whole path of the alimentary canal. Again, each of them having its own very important function, and each structure is adapted to its specific function in the digestive system. So ingestion is the taking of substances such as food and drink into the body through the mouth. Then there are two major types of digestion, which one of them is mechanical digestion, which is the breakdown of food into smaller pieces, but there is no chemical change that is happening. This only increases the surface area for the enzymes to act in chemical digestion. Then there is chemical digestion, which is the breakdown of large insoluble molecules into small soluble molecules. Other functions of the digestive system are absorption, which is the movement of small food molecules and ions through the walls of the intestines into the blood. Assimilation is the movement of digested food molecules into the cells of the body where they are used, becoming part of the cells. And then we have egestion, which is passing out of food that has not been digested or absorbed as feces through the anus outside of the body. Another process that is happening in our digestive system is peristalsis. The alimentary canal has layers of muscles in its walls. The fibers of one layer of muscles run around the canal, circular muscles, and the other runs along its length, which is known as longitudinal muscles. You can see in this picture, it shows the muscular wall and the circular wall. When the circular muscles in one region contract, they make the alimentary canal narrow in that region. A contraction in one region of the alimentary canal is followed by another contraction just below it, so that a wave of contraction passes along the canal, pushing the food in front of it. So this is how the food is pushed along the alimentary canal. This process is known as peristalsis. Now, starting with the first process, which is mechanical digestion, which involves the mouth and our teeth. These are the different types of teeth involved in breaking down larger food substances into smaller. We have the front teeth, the side teeth, which are known as canine. We have the posterior teeth, which are known as premolars and molars. This is used for biting. This is also used for tearing the food particles. These two are important for chewing. This diagram shows you the total number of teeth in each. This is the upper jaw, and this right here is the lower jaw. You can see each of these type of teeth in your mouth, mainly involved with mechanical digestion. Now, if you look at the inside structure of the teeth, you can find that the crown portion, which you can see in your mouth, has an enamel, a dentine, and the pulp. And the root portion, which is embedded in our gums and the bone, consists of the cement and the nerves. And this makes up the root. Again, eating with mechanical digestion. Chemical digestion is occurring through the alimentary canal in all parts of the digestive system. The first one occurs in the mouth, where salivary glands are found, which secrete saliva, which chemically digest salivary amylase, the enzyme, which chemically break down starch into maltose. Then the food enters the stomach, where glands in the stomach linings secrete gastric juice and hydrochloric acid. The gastric juice has pepsin, which works in acidic conditions. Where does this acidic condition come from? It comes from the hydrochloric acid secreted by the stomach, which creates a very acid pH of 2. Pepsin digests proteins into peptides. And then the hydrochloric acid also denatures enzymes in harmful organisms in food, such as bacteria. So it is also a protective acid. The food then enters the small intestine, which the first part of small intestine is the duodenum, where the pancreas, they secrete pancreatic juice and the bile. The pancreatic juice has proteases, trypsin, which acts on proteins and peptides, forming peptides and amino acids. The bile, because the trypsin has to work in alkaline conditions, bile provides those alkaline conditions. Then we have amylase, which converts starch into maltose again, and lipase, which converts fats into fatty acids and glycerol. This is a spelling mistake here. Then the food enters the second part of small intestine, which is the ileum, where the epithelial cells, there is no digestive juice produced, but it has enzymes, which is maltase, converting maltose into glucose. This maltose comes from the digestion of starch here, which is happening in the mouth and the duodenum. And the peptidase enzyme converts peptides into amino acids. This was the mechanical and chemical digestion taking place through the alimentary canal. Stomach also helps with mechanical digestion, as the movements of stomach, they churn the food and mix them with the juices very efficiently. Then we have absorption. Nearly all absorption of digested food takes place in the ileum, which is part of the small intestine, along with most of the water. Small molecules of digested food, such as glucose and amino acids, pass into the bloodstream, while fatty acids and cholesterol pass into the lacteals connected to the lymphatic system. These are all the parts mentioned here. What is happening is the ileum is very long and presents a large absorbing surface to the digested food. Secondly, its internal surface, which you can see right here, is thrown into folds. The internal surface is greatly increased by circular folds, right here, bearing thousands of tiny projections called villi. These are the villi, you can see. These villi are long and they are finger-like or flattened in shape. The lining epithelium is very thin and the fluids can pass rapidly and easily through it. The outer membrane of each epithelial cell is further thrown into folds, which are called microvilli, if you can see right here, which increase the surface area again 20 times more. The villi were already increasing the surface area and they increase it further. Then there is a dense network of blood capillaries, which you can see right here, which are tiny blood vessels in each villus. All of these structures, they aid and they make absorption efficient. Then comes the large intestine, which includes the colon and the rectum. Materials passing into the large intestine consist of water with undigested matter, largely cellulose and vegetable fibers. The human body cannot digest cellulose. And vegetable fibers, they produce roughage. There are also mucus and dead cells from the lining of the alimentary canal. The large intestine secretes, it does not secrete enzyme, but the bacteria in the colon digest part of the fiber to form fatty acids, which the colon can absorb. Bile salts are also absorbed and returned to the liver by the blood circulation. The colon absorbs much of the water from the undigested residues. The semi-solid waste, the feces or stool, is passed into the rectum by peristalsis again and is expelled at intervals through the anus. The residues may spend from 12 to 24 hours in the intestine, and the act of expelling the feces is called egestion or defecation. The last system that we'll cover is the reproductive system, which involves sexual and asexual reproduction. We will only do the difference between these two, and then we'll do the details of sexual reproduction. So asexual reproduction is the process resulting in the production of genetically identical offspring from one parent. There are no specialized cells that are involved, no fertilization or mating is taking place. Cells in one part of the body repeatedly divide by mitosis to form a structure that breaks away from the parent cell and grows into a new organism. So asexual reproduction involves only one parent, and the offspring from one adult will have the same genes because it is coming off of one parent only. They will be genetically identical. Basically, they will be clones and very, very identical to each other. The advantages of this reproduction are there is no mate needed, no gametes are formed or needed. All the good characteristics of the parent are passed on. There is no dispersal. And plants that reproduce asexually usually store large amounts of food that allow rapid growth when conditions are suitable. The disadvantages are there is no variation, there is no variety when asexual reproduction is taking place. And adaptation to a changing environment is unlikely. If the parent has no resistance to a particular disease, the offspring will also have no resistance, and there is lack of dispersal, which means lack of dispersal can lead to competition for nutrients, water, and light. Sexual reproduction, on the other hand, is a process involving the fusion of two gametes, which are special cells called sex cells, to form a zygote and the production of offspring that is genetically different from each other. Because two different cells, they fuse together, which gives them a genetically different offspring. The process of cell division that produces these gametes is called meiosis. Meiosis produces cells that are not genetically identical, have only half the number of chromosomes as the original cell. Now, half the number of chromosomes as the original cells, those are called haploid cells. And cells that have full number of chromosomes are called diploid cells. So all the cells in our body will have diploid cells because they have a full number of chromosomes. But our gametes or the sex cells, they are sex cells, or our gametes, they have haploid cells because they have divided by the process of meiosis. To restore those that diploid number, we need fertilization to take place, which is the fusion of gamete nuclei, which are the haploid cells, and then they form a zygote, which is known as the diploid cell. The zygote then grows into a new individual. Advantages are there is variation in the offspring, so they are better adapted to changes in the environment, enabling greater survival instincts. There is new variety that can be created, which may come with resistance to different diseases. In plants, seeds are produced which allow dispersal away from the parent plant, reducing competition. The disadvantages are obviously you need two parents, and growth of a new plant to maturity from a seed is slow. Again, going into the details of sexual reproduction that occurs in humans, we have the male reproductive system, its parts and its functions. The underlined ones are the important parts that you need to know. We have, this is the front view, this is the side view. We have the penis, which can become firm to insert into the vagina of the female during sexual intercourse in order to transfer the sperm. We have the prostate gland, which adds fluid and nutrients. We have the scrotum that holds the testes outside the body and maintains the temperature. We have the sperm duct, which is a tube that links the testes to the urethra. The testes, which is the male gonad that produces sperm, and the urethra, which allows passage of semen containing the sperm and also carries urine from the bladder. Then we have the female reproductive system, and the important parts that you need to know are again underlined with red. We have the cervix, which separates the vagina from the uterus, the ovaries, which contains the follicles in which eggs are produced. We have the oviduct that carries the ovum or the ova to the uterus. It is also the site of fertilization. The oviduct, most important point to remember, is the site of fertilization. The urethra, which carries urine from the bladder, the uterus, where the fetus develops, and the vagina, which receives the male sperm, which is deposited here. These are the gametes, the specialized sex cells that are involved in fertilization. The male gamete is the sperm, which is produced in the male sex organs known as the testes. They are specialized for swimming. They have the flagellum. They are produced in millions, and they are smaller, very, very small cells compared to the gametes produced in the female. So here you can see the head region, the midpiece, and the tail. The tail is only just required for its movement. The head consists of the nucleus, which contains the haploid number. It contains half the number of chromosomes, so haploid number. It has enzymes which digest the layer of the egg to enter into the female gamete. And then the midpiece contains the mitochondria, which provides the energy to the sperm for movement. The female gamete eggs are produced in the female sex organs, which are the ovaries. They are non-motile. They cannot move. They are produced in smaller numbers or a limited number, and they are large cells. They are very, very large compared to the sperm. They contain nucleus, again haploid, half the number of chromosomes, so that when the sperm and the egg meet, they can restore the diploid number of chromosomes. They contain a cytoplasm, since these are elongated cells, and you can see that these are circular cells. They have follicle cells, they have a jelly coat and a cell membrane. This jelly coat is very important because this coat is the one that is digested by the enzymes contained in the sperm cell. This is how fertilization and development takes place. So down here, you can see each stage of fertilization and development. So the sperm, they travel to the ovum. Right here, you can see sperm swim towards the ovum. Again, you can see the different layers of the ovum or the egg. The follicle cells are scattered, possibly by enzymes produced by the sperm. So the sperms, they secrete the enzymes which are digesting the follicular cells, and they then penetrate the jelly coat. Again, the enzymes in the sperm digest those. One sperm gets through the zona pellucida, the layer, again, one of the layers of the ova. The sperm passes through the cell membrane and enters the cytoplasm. Once the first sperm passes through the zona pellucida, all the other sperms are not allowed to enter the egg. Only one sperm, the first sperm, is allowed to enter the egg. The sperm nucleus and the egg nucleus fuse. This is the fertilization. When they fuse, the diploid number of chromosome is restored. So they meet, they fuse, they form a zygote. It undergoes cell division. This is where mitosis is taking place. And this is where the zygote grows into to form an embryo. Once it has formed an embryo, it travels. Now, all of this fertilization and development and this development of the embryo is happening inside the female reproductive system. The embryo travels down the oviduct to the uterus. So, like I mentioned, oviduct is where the fertilization is taking place, which is the most important part to remember, that this is the site of fertilization. From the oviduct, the embryo travels to the uterus. In the uterus, it sinks into the lining of the uterus. This is when implantation of the embryo takes place. The embryo continues to grow and produces new cells that form tissues and organs. At 8 weeks or after 8 weeks, when all organs are formed, the embryo is then called a fetus. The embryo then keeps on growing. The uterus enlarges to contain it. You can see it. This is at 5 weeks, and after 8 weeks, you can see it has formed a fetus at 10 weeks. Now, inside the uterus, the embryo becomes enclosed in a fluid-filled sac called the amnion or water sac. Again, at 10 weeks, you can see that the amnion and the water sac, which protects it from damage and prevents unequal pressures from acting on it. So the amnion or the water sac, when there's any pressure that comes from outside, this is where it's protecting it. So the fluid inside the amnion is called the amniotic fluid. Soon after the ball of cells reaches the uterus, some of the cells, instead of forming the organs of the embryo, grow into a disc-like structure, which is called the placenta. Again, this structure, which you can see, is the placenta, 10 weeks, and then again at 5 months. Oxygen and nutrients such as glucose and amino acids pass across the placenta to the embryo's bloodstream. Carbon dioxide passes from the embryo's blood to that of the mother. Blood entering the placenta from the mother does not mix with the embryo's blood. Apart from this, alcohol, certain drugs, and some viruses can also affect the baby through the mother. This is all happening across the placenta. So if the mother is consuming alcohol, this can pass through the placenta, damage the fetus. Certain drugs can affect the fetus, and viruses, for example, can affect the fetus, which most commonly causes measles or German measles, because that virus can pass through the placenta into the fetus. The placenta becomes closely attached to the lining of the uterus and is attached to the embryo by a tube shown right here, which is called the umbilical cord. The umbilical artery carries deoxygenated blood and waste from the fetus to the placenta, and the umbilical vein does the opposite, provides the fetus with the oxygenated blood and the nutrients. This right here, a few weeks before birth, you can see at 35 weeks, this is the growth of the fetus. Now, there are hormones controlling the reproduction. or the reproductive system. Now, in boys, the follicle stimulating hormone stimulates sperm production, and the luteinizing hormone instructs the testes to secrete the male sex hormone, which is testosterone. Again, we did all of these hormones and their functions and their glands in the endocrine system. The testosterone controls the development of the male's secondary sexual characteristics. These include growth of the penis and testes, growth of facial and body hair, muscle development, and breaking of the voice, where the voice becomes deeper. In girls, these pituitary hormones, again, the FSH and LH, control the release of female sex hormone called estrogen from the ovaries. Estrogen produces the female secondary sexual characteristics such as breast development and beginning of the menstrual cycle. During the menstrual cycle, there is a hormone, there is a cell or a part of that ovulation or menstrual cycle called the corpus luteum, which makes another hormone called the progesterone. Progesterone completes the development of the uterus lining by thickening and maintaining it, and it makes it ready for the fertilized egg to sink into it and develop into an embryo. So progesterone deals with the implantation of the zygote or the embryo. It also inhibits the release of FSH and LH by the pituitary, which stops ovulation. So once implantation completes, it stops ovulation so that no further fertilization and implantation can take place. Now, during the menstrual cycle in females, these are the hormone levels that are taking place. So you can see during menstruation, luteinizing hormone is very, very low. The pituitary hormones secreted are very low. Estrogen and progesterone are also very low. And this is the growth of the follicle. As the menstrual cycle finishes, these are the levels of the hormones and estrogen and progesterone. During ovulation, estrogen is at its highest, and after that, the luteinizing hormone is at its highest. Progesterone, for example, during ovulation, fertilization and development is taking place. Progesterone increases, which inhibits the release. Right here, you can see FSH and LH, so that no further ovulation can take place. This progesterone is released from the corpus luteum right here. The progesterone, for example, if no fertilization takes place, this progesterone then leads to the breakdown of the uterus lining, which is then secreted out during the menstruation outside of our bodies. Again, this little circular chart shows each cycle and the day that these processes are occurring. So 1 to day 5, there's menstruation. Then from 6th day to 13th day, you have new uterus lining that develops. On the 14th day, you have ovulation. If this ovulation does not take place, like for example, egg in the oviduct is not fertilized, it is then broken down and then it is released again during menstruation. Again, sexual reproduction brings in sexually transmitted infections known as STIs or maybe STDs, sexually transmitted diseases. It is an infection that is transmitted via the body fluids through sexual contact. One of those is AIDS and HIV. The AIDS is acquired immunodeficiency syndrome. A syndrome is a pattern of symptoms associated with a particular disease. The virus that causes AIDS is the human immunodeficiency virus. Basically, the virus that affects the immunity. It makes the immunity deficient or decreases the immunity. HIV is transmitted by direct infection of the blood. It can also be transmitted through drug users who share needles contaminated with infected blood. Can be transmitted sexually between men and women. Infected carriers that donate blood can pass on the virus, and babies born to HIV carriers may become infected either in the uterus or during birth or from the mother's milk. How can we control these STDs? We can avoid having sexual intercourse with an infected person. We can use protective barriers such as condoms by men and femidome by women. These, again, act as barriers. If a person suspects that he or she has had sexual contact, treatment must be sought at once, and both the affected person and the carrier should get treatment. Sexually transmitted infections are caused by bacterium such as syphilis and gonorrhea, which can be treated with antibiotics since they are caused by bacteria if the symptoms are recognized early enough. But HIV, which is the virus, now viral diseases cannot be treated with antibiotics, so they are not affected, sorry. Okay, here we have dealt with the major systems in the human body. Now we'll look at how the papers are designed. This is how the paper is for anyone who's attempting the Edexcel syllabus. So you have paper 1, which is available twice in a year in November and in June. They have paper 1 is a 2-hour written examination. Total marks are 110, and there are a number of different question styles, which include multiple choice, short answer questions, and calculations and extended open response questions. Paper 2, again available twice a year in November and June, is a 1-hour, 15-minute written examination. Total marks are 70, and again, the same type of different question styles. Now for Cambridge, this is the paper pattern, where paper 1 is a multiple choice, 45 minutes, 40 marks, and again, 40 multiple choice questions with 4 choices. You have paper 2, again, multiple choice, but this is extended. So core is the basic syllabus that you study. And then you have paper 3, which is theory of this core, right? 1 hour, 15 minutes, carrying 80 marks. Short answer and structured questions makes up 50% of the paper. Then you have an extended assessment, which consists of paper 2, 45 minutes, again, 40 marks, 40 multiple choice questions with 4 choices, and then theory paper 4, again, dealing with the extended syllabus. 1 hour, 15 minutes, 80 marks, short answers, and structured questions. Then you have practical assessment. You either take paper 5 along with 1 and 3, or paper 6, or you can take either of them with paper 2 and paper 4. So paper 5 is a practical test, 1 hour, 15 minutes. Questions are based on experimental skills. Paper 6 is alternative to practical, 1 hour, has 40 marks, and questions, again, based on experimental skills. Okay, since we have reached the end of our session, I can now take questions. But before that, anybody who's missed out on number or the QR code, I will just go back to that slide. So whoever wasn't present at the beginning of the session, you can always contact us. Either you can scan the QR code or you can contact us through this number. Okay, for anybody who's asking about Edexcel papers, they have a very wide, they have a lot of paper options, which we will need obviously another session for if you want to know more details about it. Otherwise, we can always provide you with the link for the syllabus specifications and the paper pattern. Okay, Protein synthesis of translation and transcription. That is a separate chapter as a whole, which deals with genetic information, DNA, how it's formed and how it's processed. So, which is not included in the systems that we dealt with today. That comes under inheritance and genetics. By the way, you guys can always unmute your mics and then ask the questions straight away. If you have any question or anything that you want me to repeat from the slides, from the presentation, I can do that. Okay, we have a question regarding vasodilation and vasoconstriction. Okay. Let me go back to the slide. So I can tell you related to the structure of the arteries and veins in the circulatory system. What is happening is, for example, you have blood that you receive directly from the aorta. So first, the vessels will dilate, right? When they receive blood, they dilate, their lumen increases. Why? Because there's an extra amount, an increased volume and an increased pressure of blood that you are receiving. So to accommodate that pressure and that volume, the vessels, they dilate. Now, after that, the pressure and the volume, the blood distributes, right? It goes further along the arteries and the veins. So the regions of the arteries and veins that it has passed through, they then come back or spring back to their original position, which is vasoconstriction, which means that it can carry low volume and low pressure. So this is basically vasodilation and vasoconstriction. Again, if I relate it to the control of body temperature, this is what is happening. Blood flows through the loops, radiating heat to the outside and cooling the body down. If the body is too hot, so what will happen is the arterioles, which are small arteries, which lead to the capillaries, to increase the blood flow to the surface of our skin so that we can get rid of the increased temperature. The blood flow is increased. How? Because the vessels dilate. Vaso means vessels and dilation means increasing in diameter, right? So to get rid of the heat, the vessels in our peripheries or near our skin, they become dilated, bringing in increased volume of blood and increased pressure. And so the heat is then taken out of the body through our pores right here. Now, once the body cools down, the vessels, they come back to their original position or original diameter. So the blood flow is reduced and the heat loss is reduced. This is called vasoconstriction. It is basically the increase and decrease in the diameter of the vessels. Any other questions? Again, if you want to connect with us, going back to this, you have the QR code, you have the number. You can always reach out to us. No questions? Again, if you want me to repeat anything, or you can just, give a feedback of how the presentation was, or if you understood everything. Oh, yes. This recording will be available on YouTube. You can always subscribe to our channel. Again, goes by the name of Academy X. I will see if I can send this presentation or the recording through WhatsApp, but this will be available on our YouTube channel. So you can always check that out and subscribe to our channel. I can also send the presentation and ask them to, distribute it among all the participants or whoever wants it. Edexcel contains all of this content, the syllabus content for Edexcel Cambridge Oxford AQA is the same. They deal with the same chapters, they deal with the same syllabus content. It is just the paper pattern that is different, and there might be a few differences. Like something that you need to study for Cambridge, you might not need to study that for Edexcel. Something that you need to study for Edexcel, you might not need to study that for Oxford AQA. But all in all, the syllabus content is the same across all these three syllabus. Okay, we have another 5 minutes to take any questions again. One thing you can do if you have less time to prepare for a paper, or if you want to cover more in a short time, is go through all the syllabus content, which is again available on their websites. Or what you can do is run through the past paper questions. The past paper questions, they usually deal with how the structures are adapted to their functions. Mostly they deal with the labelings, right? All the labelings and their parts. Even if you remember like one function of one part of any body system or organ that they're asking, that should be enough and good. Otherwise, structured questions are usually how, for example, gas exchange takes place. Now, you don't have to remember every little detail, but it is good to jot it down in points and then give your answer in points so that you remember that you have covered every point. Basic questions that they ask, most importantly, are, again, structures related to their functions or how they're adapted to their functions. Okay. I can explain gas exchange again. So what is happening is we breathe in through the nose, or let's say the mouth. The air goes through our trachea, goes into the bronchi, right? And then passes into the bronchioles. How are we inhaling? We need to create a pressure difference, right? So once we inhale, this diaphragm right here, it flattens into this position. When it flattens, it widens our chest cavity, our thoracic cavity, and the pressure inside, the air pressure inside our lungs is less. So the air can easily move into our lungs. The rib cage also expands. Once the air enters our lungs, it goes into the alveoli, the little cloud structures that we saw. Here, because we are breathing in oxygen from the environment, the oxygen gets into the alveoli or the alveolus. Now, there is a network of blood capillaries running through each alveoli. The blood carries less oxygen and more carbon dioxide. It has collected carbon dioxide from all the respiring cells. So there is more carbon dioxide in the blood and less oxygen. There is more oxygen in the alveolus. Again, concentration gradient exists. So the oxygen, which is in higher concentration in the alveolus, moves inside the blood, right here. Because the carbon dioxide is more in the blood, it diffuses and goes to a lower concentration, which is in the alveolus. Then we breathe it out. The carbon dioxide, what is happening then during exhalation is the diaphragm comes back to its original dome-shaped position, right? Again, we squeeze out the air because the rib cage becomes smaller, the thoracic cavity becomes smaller, air pressure inside increases. Why? Because the container, our thoracic cavity, is now small and there is too much air inside our lungs. So we have to expel air out. Now, the air that we expel out is composed of carbon dioxide, which is why then carbon dioxide goes from the alveoli again into the bronchi, bronchioles, trachea, and then is breathed out through the mouth or the nose. So the main site of gas exchange is the alveoli. And then inhalation and exhalation involves the rib cage and the diaphragm and a pressure difference. Okay. Okay, no further questions. I guess we'll end the session here. Again, okay, just read that. Again, I appreciate you guys showing up for this session. I hope this was helpful and I wish you all luck for your exams and your further future endeavors. Good luck, okay? Everybody take care, bye.